Good afternoon, everyone. My name is Pat Furlong, and I'm president of PPMD, or Parent Project Muscular Dystrophy, and I'm really happy that you joined us today. Today's webinar is really going to focus on Capricor. You may have or may not have heard about Capricor. This is a company that has a cell-based therapy, and we have online today Craig McDonald, who is the PI of the study. We have Deborah Ashheim, the Chief Medical Officer, and Linda Marban, the CEO of Capricor, to talk to you about this. And just let me remind you, when we talk about this cell-based therapy, we're not talking about expecting or anticipating that the cells delivered are going to be integrated into muscle. Rather, they're using, in this particular context, they're being used as delivery vehicles. This is a very exciting opportunity for this community, and we're really interested and excited about this study. So at this moment, I'm going to turn this over to Linda Marban, and we will continue the webinar. Linda?
Thank you so much, Pat, and thank you, Parent Project of Muscular Dystrophy, for giving us this opportunity to talk to the community about the HOPE-2 clinical trial. I am indeed Linda Marban, the CEO of Capricor, and I'll be talking to you for the next few minutes about the history of the technology, and then I'll turn it over to Deborah Ashheim, our Chief Medical Officer, to go through a little bit of the data that we've collected so far. And then finally, we'll finish up with Craig McDonald, our national PI, with whom we are very proud and excited to be working with in this trial. Thank you for your time again. So we are a publicly traded company, and this is a typical forward-looking statement slide. And you can see all of our filings with the Securities and Exchange Commission should you have any further questions regarding those. So let's talk a little bit about CAP-1002. And Pat, who has been with us really since the very beginning, since we started collecting some of the early stage mouse data, was correct in saying that this is a cell therapy, not necessarily a stem cell therapy. So CAP-1002 is made up of cardiosphere-derived cells. These are cells that are taken from the heart. They're a very special population of cells that we isolate out, and then we expand using proprietary techniques in a manufacturing plant so that we can take the power of these cells, which is delivered by exosomes, which I'll talk about in a few moments, that actually function, as correctly Pat said, as a local drug delivery vehicle, taking messages to cells that do a variety of things. They immunomodulate, they encourage muscle cells to re-enter the cell cycle, so encourage muscles to endogenously grow. And they also stimulate new blood vessels to form in order to feed the new muscles. And we have a lot of preclinical data that we've collected over many years, which I'll give you a little bit of a flavor of within the next few minutes. The cells have been in over 140, more than that actually at this time, people both in adults and in patients with Duchenne. And thankfully, they have proven to be very safe. So easy to deliver and a very safe therapeutic way to get the advantages of the cell therapy. So how do the CDCs work? We've spent the last decade working on understanding the mechanism of action of how these cells actually work in the body. And we've gone from treating adult heart disease, where we thought we could deliver cells and they would go into the hearts of the patients that we were delivering them to, usually patients that have had heart attacks, and that they would actually reduce the amount of scar and improve cardiac function by becoming part of the heart. What we've learned over this decade and have published in many papers is that's not how the cells work at all. The way that they work is that they go into the circulation of the body, so using any blood vessel, even a simple IV delivery is very possible. And then the cells then lodge into the blood vessels of the lungs. It's called the microvasculature. But don't worry, it doesn't cause any ill side effects because the microvasculature of the lungs literally could take up miles of ground if you spread them end to end. So it's not dangerous in terms of vascular function in the lungs. And then from there, the exosomes, which are really the active ingredient of the cells, are released. And we have a lot of biodistribution data that shows that they track to the site of injury, specifically muscle injury. That's muscles in the heart and muscles in skeletal muscle as well. So we have a dual-acting cell, muscle in the skeleton, as well as cardiac muscle, where they stimulate then this very profound immunomodulatory effect, which I'll show you some data from in a few minutes, and then leads to improvement in cardiac as well as skeletal muscle function. So as we made these discoveries, and we realized that Duchenne muscular dystrophy would be the ideal to target because it's a disease of muscle as well as a disease of inflammation, we decided that we would examine how these cells work in the MDX mouse. And actually, I'm going to start showing you data that Pat was part of the very beginning of as we began to collect this data and try to figure out how best to deploy them in this community. So shown here is some of the very early stage data that was collected when we used our cells, the CDCs. It's the active component of CAP-1002. In the MDX mouse, I think all of you in this community are very well aware that the MDX mouse is sort of the average model, animal model that we use to test therapeutics in when we want to see if they make a difference potentially in Duchenne muscular dystrophy. It's a genetic mouse that naturally gets the disease, and so we can sort of study them over time. For our purposes, because we're looking specifically at cardiac as well as skeletal muscle function, we age our mice, and we wait until they have evidence of cardiac disease, as would be typical in the later stage patients in this disease. And then we treat the mice, and we can examine whether the cells mediate a beneficial effect. In panel A on the slide that you're looking at, you can see three groups of animals. There's a light gray bar on the top. Those are normal lab mice, and we use those sort of as controls to make sure our experimental conditions work. And then there's two groups of animals that you can only see the difference of once the lines begin to separate. And those are the MDX mice that are randomized, just like people would be, whether they receive cells or receive a placebo. And what you can see here is that the animals show an improvement in cardiac function that begins to wear off about 3 months into the therapeutic. And then we can give another injection of the cells, and we see a synergistic improvement in function again, and that's in cardiac function in the mice that are treated. Whereas the standard MDX mouse who got the saline placebo continues to decline. It gets worse over time and would be reflective of potentially what we would see in DMD, which is a steady, slow decline in function. The other observation we made, which was actually game-changing for this therapeutic, is shown in panel B. So we took the same three groups of animals, standard normal lab mice, just to make sure experimental conditions aren't crazy in some way or another. And then we took the MDX mice and again randomized them, and you can see that at time 0, they all start from the same baseline ability to run, and this is treadmill running that we're looking at. So it's kind of the mouse version of the 6-minute walk test. And again, we see a similar pattern, which is that we see an improvement in the mice's ability to run over time, which begins to decline again after about 3 months. And even though the scale of this graph is pretty large, it's actually almost a doubling in the mice ability to run. So what we realized is that we had dual activity of our cells. They were targeting both cardiac muscle as well as skeletal muscle. And so we decided to look a little bit further and see if the skeletal muscle function was in fact related to improvements in actual muscle ability, or whether it was somehow related to the improvement in cardiac function. And so the next set of experiments that we did is we took the same, again, 3 groups of mice, and we treated them exactly as we talked about, the standard normal lab mice, and then the 2 MDX mice groups. Only difference was one got placebo and one got cells. And then we looked at isolated skeletal muscle force. So what we were able to do is you take out a skeletal muscle. It's called a soleus muscle in this situation, which is like a leg muscle. Could be like a quadricep in a person. And you put it into a muscle bath, and you stimulate it to contract. And what you can see here shown in these bar graphs is that the animals that got the vehicle did much worse than those that got the cells. In fact, there's no difference in the animals that got the cells in their ability of their skeletal muscle to contract, and 3 different measures of contraction when they were treated with the cells, whereas those that got the vehicle were statistically less than the wild types, and also those that were treated with cells. So we know for sure that there's an independent skeletal muscle impact on these cells. Okay, so how do we know that this was making a difference? Well, we did a significant amount of preclinical work. The data has been published in a paper in a journal called Stem Cell Reports, where we studied the effect of our CDCs, CAP-1002, in mouse and human models of DMD. We're happy to provide access to this paper, or you can find it easily online. And what it basically shows is that you can take the downstream effects of the dystrophin deficiency, which we all well know is damage to the cell membrane, increases intracellular calcium in a very pathologic way, which then leads to all of these downstream effects: myocyte loss, oxidative stress, inflammation, damage to the energy-producing mitochondria, which is how the cells give enough energy for muscles to contract, and is very, very important in normal cellular function. Microvascular ischemia, which means that the blood vessels don't get enough blood flowing through them. And then finally, all of these things lead to cell death and fibrosis. And what we show is that the cells and CAP-1002 are able to target all of these downstream effects and mediate their beneficial effect, leading to the functional effects that you just saw a few moments ago. So we believe that there is a lot of room in the toolbox, colloquially, to treat Duchenne muscular dystrophy. These are exciting days for you and your community, and I am amazed to sit by and watch the power of the parent lobby and people like Pat, PPMD, to really be able to make a difference in getting attention to this orphan disease. But now it seems like there's gene therapies that are going to be working. They're actually going to be able to do some repair of the dystrophin deficiency, and over time really be able to make a significant difference in this disease. But let's remember that the disease itself is still present, and therefore you're still going to have inflammation, you're still going to have fibrosis, you're still going to have the energetic effect, and a variety of other symptomatology of this disease that will need to be addressed. And so we think that CAP-1002 fits very nicely into that specific paradigm and is able to work in conjunction with the gene therapies so that we provide the best possible opportunity for the boys and young men with Duchenne to have as normal a life as is possible. So you'll hear in the next few minutes from Deborah Ashheim, our Chief Medical Officer, about HOPE-1, and you'll hear why, again, it fits a niche that is not being filled by many other therapeutics, where we're going after and providing opportunities for those patients that are in later stages of the disease process, which also gives us a unique place in the toolbox to treat Duchenne. So with that, I'm actually going to turn it over to Deborah. Thank you for your time, and I look forward to answering your questions now or at any time in the future. Deborah?
Thank you, Linda. So I am Deborah Ashheim, as you heard, and I'm going to talk to you about the HOPE trial, which was the first trial that we did, taking into account all that preclinical work that led to the summary that Linda just provided to you, to start to evaluate CAP-1002 in patients with Duchenne muscular dystrophy. Now, bear in mind that Linda mentioned that we started by developing CAP-1002 to improve cardiac effects, although we got quite interested in the potential for it to impact the skeletal muscle. But at the stage of the game that we were at when we initiated the HOPE trial, that first trial, we were still directly administering the cells into the heart as what we were assuming was the primary organ that they were going to impact. So the HOPE trial was designed as a safety trial to evaluate administering these cells directly into the arteries that go to the heart, the coronary arteries. It was a one-time infusion, and patients were randomized, like a flip of a coin, the way you are fairly used to at this point in time with the number of clinical trials that are ongoing in DMD. They were randomly assigned to 75 million cells, that single one-time infusion, or usual care. And then at the end of the trial, the usual care arm was assuming that the Data and Safety Monitoring Board, this independent board that reviewed the data, as the safety and effectiveness data as it was presented to them several times during the trial, made a recommendation to move forward to the open label extension. So the trial was conducted at 3 sites. The main site was at Cincinnati Children's, and the other 2 sites were at Cedars-Sinai and at University of Florida in Gainesville. Patients were as you would expect, because we were targeting patients with cardiomyopathy, so with the heart disease related to Duchenne muscular dystrophy, and fairly extensive involvement, these patients were older than most clinical trials. As you can see from this slide, the average age, the mean age of the participants in the trial were about 18 years old. And as you can also see in this slide, a majority of them were non-ambulant by the time they enrolled in the trial. Again, since we were defining the population, they needed to have scar in at least 4 segments of their heart by MRI at baseline, because that was primarily what we were looking to see. Could we affect the amount of scar in the heart? And so, not surprisingly, this was a slightly older group than usual. In fact, what we did see was that we could impact the amount of scar in the heart. After that initial infusion of CAP-1002, we did a host of assessments over time. There was, as you saw on the last slide, an interim analysis at 6 months and then a primary analysis for the final results at a year. That was the final efficacy results at a year. And what we saw, and you can see in the panel on the left at the bottom of this slide, is that in the blue boxes, these are the participants who were randomly assigned to the usual care group. So they continued to get their optimal standard care from their doctors as well as from being followed at the research center. The amount of scar in the heart increased, both a modest amount at 6 months, which you wouldn't expect it to be too much by 6 months, and then more by a year, which is what you would expect for the natural history of the disease. What we saw in the red box, and you can see each of these little dots actually represent one of the participants, because, you know, since it's a small group of participants, it's sometimes easier to see on an individual level changes over time. So what you see in the red box is that at 6 months, in fact, there's a little bit of a decrease, which actually is fairly meaningful. It's almost statistically significant at 6 months. And at a year, the decrease in scar was significant. So that was really reassuring to us. What we then wanted to understand, though, is what effect, we know that more scar, it does bad things. Worse outcomes are associated with progressive increasing amounts of scar. But because there hasn't been a therapy that actually reduces scar, meaningfully reduces scar, nobody's really been able to look at what's the clinical impact and what's the physiologic impact of actually diminishing that scar. So we looked at what's called regional wall motion, the regional function of the heart muscle, to see whether or not in the areas of the heart that were most affected by scar, which, by the way, were the areas of the heart where theoretically we were expecting that a reduction in scar might have the biggest impact, would that actually translate to a better ability to squeeze, for that muscle to squeeze? And what you see in the middle panel and in the right panel is, in fact, that is what we saw. So in the middle panel, we're looking at the inferior wall of the heart, which is the first area of the heart that's affected by scar in DMD. It's a very classic pattern, which is unique to Duchenne heart disease. It starts in the inferior wall and then progressively moves to the anterior wall, the lateral wall, and then the septum. And so the most affected area is the inferior wall, and then the second most is the anterior wall, et cetera, et cetera. So what we see here is that, again, the blue, the usual care patients, they actually had a little bit of a reduction in their ability for the heart muscle to squeeze in that inferior wall at 6 months. And around a year, you know, it's sort of essentially hovering near unchanged. What we saw with the patients treated with CAP-1002, though, was that there was an improvement in the ability for that heart muscle to squeeze in the inferior wall. actually met statistical significance, which in this very small group of patients was really quite reassuring. And I should also add that these analyses were done by a core lab that was blinded to the treatment assignment, meaning they didn't know what each of the participants received, whether or not it's usual care or placebo, and they had no information about how those participants were doing over time either. They simply had the images to analyze. And
Deborah, it's Pat. I just have one other question. Did that translate into a change in the ejection fraction?
Thank you, Pat. So that did not translate into a change in the ejection fraction. I have to say I would have been stunned if it had, bearing in mind the fact that this is a very diffuse process, the scar and therefore the damaged heart muscle, certainly at the point at which we were infusing it, and it's a single infusion that we were given. So at this point in time, we saw just areas of improved function in each region, but the biggest area of improvement was in that inferior wall. And then as you marched out to the other areas, say the septum, there's a whole lot less involvement there until really the end of the disease. And so there was less impact on the heart muscle function there.
But you're expecting, or we might anticipate that in a study where there's repeated infusions of these cells and over a longer period of time, that we may well see a change in the ejection fraction.
Yes, we do think that is.
Yes?
Yeah, we do think that is possible. Now, as a heart failure cardiologist myself and a clinical trialist for a lot of years, I can also add, since I've been working on therapies for other types of heart failure for almost a quarter of a century, that changes in ejection fraction, although nice to see, are not necessary to see for actual real improvements for a heart failure therapy, for heart failure therapies that are on the market that actually have been doing wonderful things to people. So although, yes, I do think that we may be able to see a change in ejection fraction, I have to say that I won't be too disturbed if we see all the other good things and we don't see a change in ejection fraction.
Okay. Well, at least you see a stable ejection fraction.
Yes, exactly. Exactly.
Thank you.
Sure. Okay. So we also looked at a host of a lot of other parameters, skeletal muscle, et cetera, which is not surprising given the preclinical study, the mouse studies that Linda spoke to you about earlier. We wanted to understand in patients, in humans, whether or not we might be able to see some of those effects as well. So we looked at the 6-minute walk test. Given the fact, as you saw a couple of slides ago, that really most of our participants were non-ambulant. In fact, there were two patients in each of the groups who were ambulatory, and other than that, everybody was non-ambulant. So 6-minute walk test, we didn't even bother to analyze. It was not helpful. Skeletal muscle function we then assessed using the upper limb, because obviously that you can evaluate over the spectrum of non-ambulatory patients and ambulatory patients. Again, for you as a group, you probably could give me a lesson in the performance of the upper limb and its significance. And certainly Pat and the PPMD team have been instrumental in sort of the collaborations amongst the community and the doctors, physical therapists, clinical trialists who developed the poll with you in order to identify a clinical assessment that was as meaningful as possible to the types of activities that are really critical to activities of daily living for you and your sons with DMD. So with the poll, as you know, it's evaluated in 3 different levels: the shoulder, the mid-level, and the distal level. In the patients that were enrolled in the HOPE trial, again, because it was really quite advanced disease, a vast majority of them had already lost shoulder function at baseline, at the time they participated in the trial. So, there was, if there's no ability to test it at baseline, then there's no ability to look for a change. So that was not particularly useful in terms of analyzing data from this particular population, which is why what you'll see here is analyses related to the mid-level, so the elbow level activities, and distal hand and finger level activities. And what we saw was, again, the blue bars are the participants who were randomized to the usual care group. There was not a lot of change over a year, at week 6, month 3, month 6, or month 12. Sort of hovered around no change. However, what we did see in the patients who received the CAP-1002 directly into their heart was that there was an improvement in the PUL. When you look at this, this is showing you a percent change from baseline. So at 6 weeks and at 3 months, you see the biggest improvement. By 6 months, it's really pretty much back to where it was to begin with. Which, if you remember, the preclinical studies from a time frame is pretty consistent with what we saw in the mice. It sort of wore off. If you remember that slide that showed the figure that showed the effect on the heart muscle function, by 3 months when they re-injected, most of that effect had worn off. Now, there is some signal at 12 months here, which, at this stage of the game, we can't completely understand. We can't completely explain, bearing in mind that this is, for this trial, out of necessity, was not a blinded trial, so it's a little hard to understand it. However, in the context of the fact that we still saw a change in the scar in the heart and a change in the regional wall motion in the heart, there probably is still a signal, something going on out at month 12. But bearing those time frames in mind, really what we concluded from the HOPE trial is that CAP-1002 was safe and well tolerated. There were some adverse events that were associated with the administration of the cells into the coronary arteries themselves, so catheterizing a heart, which I should pause and give a shout out to the participants in this first HOPE trial, because they really were brave pioneers, particularly since and their families, since this is not a procedure that's routinely done in patients with Duchenne, and in fact most of you are told not to do because there haven't been therapies to do it. But at that point, since we didn't have the additional information about whether or not the cells would be effective enough systemically through a peripheral IV, we clearly had to start with administering them into the heart. So they were not, serious events, but there were events associated with the infusion procedure. But on the whole, it was safe and well tolerated. On the upside as well, we did see some early signals that there is a benefit, or that there is likely to be a benefit with CAP-1002 on both the heart in terms of the scar and the ability of the heart muscle to squeeze regionally, as well as the skeletal muscle. Now, what we also learned, which we were not surprised about, is that we would likely need to do repeat dosing in order to see a potential sustained effect. And with that, those are really the tenets with which we designed, in collaboration with a number of people, including Pat and Craig McDonald, who you're about to hear from, and a number of Craig's colleagues. We designed the HOPE-2 trial. And so now we are thrilled to be able to start discussing more broadly with you the HOPE-2 trial, which began fairly recently, and Dr. McDonald's site, as the Vanguard site, since he's the PI, were the first out of the gates. So he's going to talk to you about the trial design itself, and can give you some additional pearls of wisdom since they now have the most experience to date.
Again, and just before you start, Craig, I'm just going to kind of back up and say and ask a couple questions. One just to clarify, in the trial that was just described, in the delivery of these cells via cardiac cath, it was only a single one-time injection for these cells. So there weren't repeat injections. It was a single one-time cardiac cath utilized for the delivery of these cells. And second, also, Deborah, there is a question about when you're talking about adverse events, you're talking about the abnormal rhythms that were associated with the delivery of these cells. So could you just speak to that before Craig goes on?
Sure, happy to. Yes. So the most common thing that we saw, and it wasn't in everybody, but it was in almost 40% of the participants who had the catheterization that Pat was just reminding you about and the infusion of the cells, was an abnormal heart rhythm. So either a slow heart rhythm, atypically slow for that person, or irregular atrial fibrillation. And what would happen when we saw the atrial fibrillation or the slow heart rhythm is it was very consistently in the same vessel of the heart. We would see during the infusion that abnormal heart rhythm, and then if you slowed or stopped the infusion, that abnormal heart rhythm would stop. Now, the patients were not aware that they were having that abnormal heart rhythm. They weren't feeling anything, but you can see it on the monitor, and it definitely tells you that something in there is getting tickled, and acting in this atypical way. I can also tell you that there are, specific to Duchenne cardiomyopathy, there are reasons to believe that it's probably related to a combination of the underlying heart disease related to the Duchenne, because you actually can see these atypical heart rhythms. But clearly, we were, by having a catheter in there and infusing a liquid in there, we were clearly triggering it with that infusion. It would stop as soon as the infusion stopped. And I can also tell you that we did a lot of cardiac monitoring for both groups of patients, the usual care patients who didn't have the procedure and the cells, and for, obviously, the patients who did have the cells at baseline and then at a number of different time points throughout the rest of the trial. I can tell you that both groups of patients, and actually this is another publication that we're working on with the investigators from the original HOPE trial, because I think this is very important information to get out there. But both groups of patients had just small amounts, but different types of abnormal heart rhythms over the course of the trial. And the patients who had the abnormal heart rhythms during the infusion didn't have any other sorts of events associated with them. No chest pain, no problems after the catheterization, no problems during the follow-up period that were at all related to this.
Right, thank you. So
thank you, Deborah. Thank you, Pat.
The abnormal rhythms were only present during the infusion itself, correct?
Exactly.
And once that catheter was pulled, there were no further abnormal rhythms.
Yes, related to the procedure, exactly. Yep.
Great, thank you. Okay, thank you so much.
So, Pat and Deborah, this is Linda. Can you hear me okay?
Yes.
We can, yes.
Okay. So I'm the CEO of the company, but I'm also the functional chief science officer. I'm the person that bridges things from bench to bedside and have spent my career doing that. And one of the connections I'd like to make here, and thank you, Pat, for your questions and Deborah for your answers, but the good news is in HOPE-2, none of these issues will be real because one of the things we've been able to do at the bench over the past 2 years is realize that we don't have to catheterize the coronary arteries of these patients to deliver these cells and get the beneficial effect. As you heard me say a little bit ago, we learned that the cells are functioning as a local drug delivery system to get to the site of the injury by releasing the exosomes that travel based on signals that are sent out from injured tissues. And so we can use a simple IV delivery method, which is what's going to be done in HOPE-2. So really, this is exciting for the community and very exciting for us as well, because we're able to take the beneficial effect and actually do it in a way that there should be none of these cardiac rhythm events from putting a catheter into the heart. So, of course, if this was life-saving for boys or young men, the risk was very, very small, and rhythm disorders were gone by the time they left the cath lab. So it's really something that, not would be a long-term issue. But nevertheless, we've been able to sidestep it completely by going backwards into a IV where we won't be putting a catheter or liquid directly into the heart.
Right. Thank you, Linda, for that. And I think that's really important, but I think it should not go unnoticed by our community, the fact that the earlier trial does give us insight into cardiac cath in a Duchenne patient, and should there ever be a time when another therapy complementary to yours would be given or considered and the requirement direct delivery to the heart, you have really broken ground in that area and really, really informed us about the opportunity, what challenges might exist, and how to do it. So I just didn't want that piece of work to go unnoticed here. So thank you.
Thank you so much, Pat. That's terrific.
So with that, I think we can turn it over to Dr. McDonald to discuss the now ongoing HOPE-2 trial.
Great. Can you all hear me?
Yes, great. Thank you.
Great. So first of all, just as a matter of disclosure, I am the PI of the Capricor trial, and I do consulting work for Capricor as well as a number of other companies in the Duchenne space. This trial really is very exciting to me because first of all, this therapeutic really hits multiple key targets to ameliorate and improve the pathogenesis of Duchenne muscular dystrophy. And I think that with the number of targets here, just as an example, it's not just immunomodulatory, antifibrotic, pro-regenerative, but also we're seeing, at least in the animal data, improvements in the bioenergetics of the muscle fibers, the cellular energy. And you have to have the metabolic machinery in order for a muscle fiber to regenerate. And so I think we see a number of Duchenne therapeutics right now that are actually focused on mitochondrial targets. And I think what was also exciting to me about the data that Deborah just shared with you all is that we also saw some nice correlation between the improvement in cardiac scar and wall thickening and the improvement in the pulse. So the patients that actually did show improvement by MRI and cardiac scar and wall thickening were the same patients that showed the most improvement in the upper limb function, which to me argued for really a systemic mechanism that we see with these local drug delivery vehicles, the exosomes. So I'd like to get into the actual HOPE-2 trial. If we could go.
And Craig, I'm going to stop you just for two explanations. For those on the call, they might not understand completely what immunomodulation is and mitochondrial biogenesis. So can you just give us a 2-minute biology update on what are those processes that are occurring and why would you target them?
Well, first of all, with regard to the mitochondria are really the energy factories in the cells. And we know that in Duchenne dystrophy, for a cell to regenerate, you have to have healthy, functioning mitochondria to produce the ATP and produce the energy in order to allow those regenerative processes to occur. And when you have a leaky membrane due to dystrophin absence, what happens is calcium tends to leak into the inside of the muscle membrane, and that calcium is actually toxic to mitochondria. And what you get as a result of that is you get excessive increases in reactive oxygen species, which are toxic. You also get a decrease in energy production. So there's been a lot of work done in Duchenne dystrophy that shows that if you can actually improve the health of mitochondria, you can actually improve the regeneration of muscle fibers and reduce the pathology in Duchenne dystrophy. And in fact, when you look at therapeutics such as adebenone, which again, the Santhera is running the adebenone trials, that has a mitochondrial target. Astellas actually has a mitochondrial agent, and then there's also mitochondrial agents that target mitochondrial biogenesis. In terms of just one example of an immune modulatory pathway, when you get inflammation, you knock down these microRNAs, which actually impact the expression of dystrophin. And in fact, CAP-1002, the animal data really shows that, for instance, you can actually have a positive impact on these microRNAs that actually would theoretically lead to improvement in dystrophin expression. So I think that gives us hope that perhaps this therapeutic could be synergistic with other dystrophin restoration strategies used in Duchenne dystrophy.
Thanks, Craig.
So I'd like to again get into the study design here of the HOPE-2 trial. This trial really, in my mind, is a historic trial. It's really the first systemic cell-based trial in Duchenne dystrophy. The myoblast transfer trials of the 1980s and were really injectable trials, but this is actually a systemic cell-based trial. The trial is actually going to be enrolling 84 patients with documented Duchenne dystrophy, ages 10 and above, with documented upper limb impairment. These patients will all be on stable steroid regimens and have at least cardiac function with ejection fractions greater than 35%, as well as forced vital capacity greater than 35%. That's really more of a safety inclusion criteria. And these patients will be randomized 1-to-1 to receive CAP-1002, and as Linda and Deborah mentioned, the patients will be getting systemic injections through a peripheral IV of 150 million cells. This is really the largest dose of CAP-1002 that's been used in humans. We've already dosed 3 patients who have tolerated the first dose well. It's 1-to-1 randomization, so 50% of the patients will be randomized to placebo. The plan is for those placebo patients to ultimately get access to the therapy after they've completed a 12-month period of follow-up through an extension phase. And then this isn't too onerous a protocol because the infusions take place every 3 months. So many patients do travel to the clinical trial sites. There's also some visits that occur in between the infusions. So the infusions, for instance, are occurring at baseline, and again for the first dose. The second dose is given at 3 months. There's another dose given at 6 months and a dose given at 9 months. So 4 doses total. And then there's additional laboratory visits which actually can be done in the local community, which occur, for instance, at week 6 and at month 4.5. If we could go back to the previous slide. I just want to discuss the endpoints. So there are a variety of safety endpoints that are going to be evaluated, as well as efficacy endpoints. And the other historical aspect of this trial is that this is really the first registration trial to use an upper limb endpoint, the performance of upper limb, as a primary endpoint in a Duchenne trial. And so the primary endpoint, again, is the performance of upper limb. There's a number of pre-specified safety endpoints. There's also secondary endpoints, which include the performance of upper limb measures at 3, 6, and 9 months. We'll get important information in terms of the time course of the response. Other secondary endpoints include cardiac function by MRI, and then there's a number of exploratory endpoints, which many of you are familiar with, elbow grip and pinch strength, as well as pulmonary function testing, the North Star ambulatory assessment for those ambulatory patients, blood biomarkers, quality of life measures, and resource utilization. So the patients that are being targeted in this trial are actually late ambulatory patients as well as non-ambulatory patients with upper limb impairment. So these aren't the typical patient population that are being targeted by Duchenne, most Duchenne clinical trials. So the inclusion criteria for this trial is, again, genetic confirmation of Duchenne dystrophy. As evidence of reduced upper limb strength is measured by the performance of upper limb measure. And if you think about that, it's really patients who have some impairment of full overhead reach are the patients that are really being targeted for this clinical trial. Those are patients we expect to see changes over the course of 12 months. And those are actually the patients where we actually saw improvements in upper limb function, which I think was really unprecedented in the first HOPE trial. Also, patients, if they are ambulatory, should have reduced ability to walk and run. That'll be measured by the 10-meter walk-run test. So these aren't going to be a patient population, for instance, with 6-minute walk distances between 300 meters and 450 meters, which are being targeted by many of the dystrophin restoration trials. Patients who are adult patients, again, this is not limited although we're focused on patients 10 years and above, we're not limiting our enrollment to pediatric age groups. There can be adult patients in the trial, but the patients who are adults in the trial need to show evidence of loss of independent ambulation by the 18th birthday. Also, all patients will be on systemic steroids for at least 12 months, with stable dose for at least 6 months prior to entry into the trial. And weight and side effect adjustments and dosing are allowed for study entry. If we can go to the next slide, these are the exclusion criteria. We purposely are trying to avoid patients with impaired cardiac ejection fractions less than 35%, and also patients with forced vital capacity less than 35%. That's more just for safety considerations, and also just because of issues with regard to insulin resistance and perhaps dosing questions. We're excluding patients that have significant increases in their fat tissue as measured by body mass index greater than 45. Another exclusion is two particular mutation constructs. One, exon 44 skip amenable patients, where at least 50% of those patients have been shown to ambulate as late as 18 to 20 years of age and have slower disease progression. And then those with deletions that are isolated to exons 3 to 7 are exclusions. The hope is to actually conduct an additional parallel trial specifically in exon 44 skip amenable patients to actually ask some additional research questions. And there is a hope to conduct a parallel trial. Also, FDA-approved Duchenne exon skipping therapy can be utilized, such as Exondys 51, if the patients have been on stable doses for periods of greater than 2 years. So an exclusion would be a patient who's just recently started Exondys 51. Weight-based dose adjustments are allowed. Human growth hormone administration within 3 months is an exclusionary criteria. A debenone within 3 months, and then a cell-based product within 12 months, or an investigational product within 6 months, or other exclusionary criteria. If we could go to the next slide. trial is now open for enrollment. We've enrolled patients here in Sacramento, also University of Utah, Cincinnati, Children's Hospital of Colorado, and Washington University St. Louis are now open to enrollment. We expect other sites to be opening in 2018. If we could go to the next slide. Again, other considerations that really both ambulatory and non-ambulatory boys and young men really could be eligible for this trial. And I think this really is a historic trial because we're targeting largely a late ambulatory and non-ambulatory population with upper limb impairment, which have really been underserved by clinical trials. And these patients, this is really, I think, a unique trial because we are targeting this underserved population with a therapy that we hope produces improvement or stabilization of upper limb function. Long-term eteplirsen use is permitted. The center visits are approximately every 3 months. The infusions are every 3 months. It's not as onerous a trial as many that we are involved in Duchenne dystrophy. And there's a fairly robust travel policy to reduce burden on the participants and families. And also open label extension to offer CAP-1002 to participants who are randomized to placebo for 12 months. So the clinical trial contact information is provided also on this slide. And I think with that was really the most of the comments I wanted to make, and I think we have a few minutes now for questions. We just want to thank the original HOPE study participants and the HOPE-2 trial participants and their families, and also thank the multiple foundations which have provided funding for this trial. I think this is a really exciting time. We're going to learn a lot about trials in non-ambulatory patients with upper limb impairment. Through this trial, we're going to learn a lot about the endpoints through this trial. So I think this is really a very exciting and historic trial.
Thanks, Craig. That was really helpful. And I agree with you that this is a really very novel and important trial because of the population it's serving, and also because of the use of the pool as the primary outcome measure, which should give us a lot of information about the pool as well in terms of as an outcome measure that's really useful, and the ability to include then a patient population that's non-ambulatory. Some of the questions that have come in is around mutations. The specific exclusion criteria for this is exon 44 skippable, as well as the 3 to 7. So those of you who have deletions that are different from 3 to 7 mutation or different from the exon 44, those are inclusive in the trial. But if you don't mind, Craig, I'd like to go back to slide number 19. So that is about the exclusion criteria, let's start in the inclusion criteria. That you're right to go back to 18, and let's talk about that. In the inclusion criteria, there's no mention of cardiac drugs. As you know, many of these children are either on losartan or an ACE inhibitor. Some of them to include eplerenone. So I'm assuming that those are not, or those are certainly acceptable within the trial as long as they're steady state. Is that correct?
Yes, absolutely, and many of those are really considered standard of care, so all of those are acceptable.
Good, thank you. And I guess one of the things that I didn't hear you mention is we talked about earlier the first trial, the HOPE trial, and with a cardiac cath. So are those patients eligible to do this HOPE-2 trial, and under what circumstances would they or would they not be included?
Yes, they actually are all eligible to participate in the HOPE-2 trial. Potentially one of the advantages is the larger dose because this is a larger dose being administered in the HOPE-2 trial. But there's also there is a extension phase protocol that also the HOPE-1 patients can elect to participate in, as being administered at those original study sites.
So in the extension study for the HOPE-2, that they would be given, I'm assuming that if you're in the extension study, you're going to be given 4 doses of the CAP-1002. Is that accurate?
Yes, through an extension protocol, the placebo patients rolling into an extension protocol would be given 4 doses of the CAP-1002. Was that your question?
Yeah. And those that complete the study, that are in the HOPE-2 study, Craig, and once the study concludes and the analysis is being done at 12 months, are you all planning? I know you are planning then, hopefully, to see positive results, and then, as this is a registration trial, go for a decision within the FDA. But in the interim of that analysis and the FDA decision based on hopeful positive results, there is a lag time. Is there a consideration of, especially with those people, the individuals who participated in the study, to continue to dose them at 3-month intervals until there's a decision?
Yeah, I think that's definitely a topic of active discussion right now. I think it would be optimal to do that. I think it's critical to actually raise the funding and resources in order to do that. And obviously, if we're seeing positive signals from the trial, it's going to make it a lot more compelling to keep these patients on the therapeutic longer term.
Absolutely. And you mentioned as well and I know I'm not following the slides, but in the sites that are currently open, you've mentioned those. But how many more sites are going to be open? Are they geographically distributed across the country, and will we see sites opening in the East? You've got stars there without identifying them. But how do you see as these sites open up, and what is the timeline for the sites, for instance, that I see on the East Coast as well as the South in Florida, as well as in the West?
Yeah, we're really hopeful that the vast majority of the sites will be open by late summer to early fall, and I think there is a good geographic distribution. I think we've certainly targeted sites with geographic distribution in consideration, as well as sites that really provide, I think, state-of-the-art, excellent Duchenne care. So I think we're probably ultimately going to see 10 to 15 sites participating in this trial.
Yeah, thanks. So many of the sites that you're identifying here are the Certified Duchenne Care Center sites, so they certainly do and will provide optimal care along the way. One of the questions is about international participation from Canada or from outside the U.S. Is that possible, and what would the requirement be?
I think at any study site, we're always open to international participation, but it really, a lot of times, it really depends on a support network that's available, within the U.S. or in proximity to the site, either a relative or a friend, or on that basis, and obviously documentation of mutation and the types of historical information we would need to determine eligibility for the trial.
Right,
but I think sites are certainly open to at least evaluating patients from outside the U.S.
Right, and while there's a great travel reimbursement, it doesn't mean living in the United States. So you're going to have to be traveling. I think the other question about international participation is that the young person that would be participated in the study needs to be able to speak English and follow directions and sign an informed consent. So those are some of the criteria as well. I think on a different topic, and this is just the design of the trial, Greg, you mentioned a 1-to-1 placebo. Many of us who are used to seeing these trials see a 2-to-1 placebo, and as we all know, those of us who are parents really are reluctant to or wish that we wouldn't have our sons exposed to a placebo. Can you speak to the rationale behind the 1-to-1?
Yeah, I think there's always there's always advantages and disadvantages when you talk about a 1-to-1 randomization ratio versus a 2-to-1 randomization ratio. One of the issues with the 2-to-1 is it takes longer for the trial to enroll, and so therefore it takes longer for the data to become unblinded. And I think we all want to see data that's meaningful as soon as possible so we can get this therapy perhaps approved if the data looks compelling, or if allowing patients to really move on to other therapeutic options in a very timely manner. And I think the longer a trial rolls out in terms of requiring a longer period of time for enrollment, the longer that data continues to be blinded, and the more you're actually delaying access to other therapeutic options. And so I think we all want to get to the correct answer really in the most efficacious and efficient way possible. And that was really the rationale for the one-to-one randomization. And then I think there's also economic considerations as well. This isn't big pharma running this trial. This is a really committed company that has a really exciting therapeutic, but they obviously have their own resource challenges.
Yeah, sure. Thank you, Craig, for that. That's a great explanation. You mentioned idebenone as part of the exclusion criteria. Were you speaking of idebenone and perhaps other drugs such as ubiquinol or others that might be involved in these children's care, these young men's care? Is it steady state greater than 3 months, or is idebenone an exclusion criteria completely, and are there other drugs that would be in the exclusion criteria regardless of the length of time the individual's been taking it?
Yeah, the idebenone actually there's a washout period, so it is an exclusionary criteria. Obviously, if somebody's in the idebenone trial, that would be an investigational product, and they wouldn't be eligible to enroll in this trial. But if perhaps they were taking idebenone through an online source, idebenone can have impacts on the mitochondria, and we don't want a situation where we have a number of patients, young men who perhaps were on idebenone who get randomized to the placebo group and continue to take the idebenone. So that was really the explanation for idebenone specifically. There are other drug considerations which I would leave that up to the site investigators to go over with the families. But these are the
right. Thank you.
We talked about.
So some of these questions are really going to be an individual discussion with this site that's screening you, and they may have other questions that go to Craig or others in the company. I think just reiterate, Craig, the exon 44 skip amenable and the 3 to 7 are often individuals who have a slower progression of Duchenne. So in choosing to keep those particular mutations, not outside those mutations, but those particular exon 3 to 7 or a 44 skip amenable patients, because of the potential for a slower progression, you're trying to keep the population you're recruiting pretty Duchenne natural history population so that you can, at the end of the day, more quickly see the results of benefit versus others. Is that right?
Yeah, absolutely. And there wouldn't be any reason why those patients wouldn't benefit from the therapy if it were to become FDA approved. We have additional scientific questions to ask, for instance, with the exon 44 population. So we're in the process of trying to work out a parallel treatment protocol, for instance, for those exon 44 patients. That's an active area of discussion right now. But it really had to do with the ability to power an 84-patient trial over a 12-month period of time and get an answer and avoid a failed trial because of a large number of patients, for instance, getting randomized to one group versus another.
Thank you. And you mentioned that eteplirsen-treated patients for longer than 2 years are included in the study because obviously we in this community and you, Craig, and all of us want combination therapies. There's a mention of the potential of benefit for these cells, CAP-1002, to have benefit in expressing dystrophin, so other exon skipping studies or the gene therapy microdystrophin studies But at this moment, we can't combine this particular drug with any other drugs because it's investigational, and so are the microdystrophin studies investigational. So should this be approved, then there could be a discussion of whether or when patients on other microdystrophin studies could perhaps access this therapy. But I think that's looking a little too far forward. Would you agree?
Yeah, I think that's looking a bit too far into the future. But I think the important point is, and that, if you're on an FDA-approved therapy on a stable dose, it takes a while for dystrophin to be produced. But patients who've been on the therapy for 2 years or more are perfectly eligible to be in this trial. And we fully anticipate that there might be positive synergistic effects between CAP-1002 and some of the dystrophin restoration strategies.
Yeah, and that certainly can't come too soon for any of us, as you might know. I think, Craig, this, for the most part, wraps up our questions for this particular study. I can't thank you enough. One last question is, can you give us a timeline for the rest of these sites? Do you expect them all be up and running by fall, or do you think the timeline's a little bit longer and toward the end of the year?
Deborah, do you have a sense? I'm certainly hopeful that we're going to see the requisite number of sites up and running in the fall. But,
yeah, in fact, we at this point in time, it looks like we really should have them all up and running by early fall.
Good, thank you.
Yeah, we're helping them out as best we can, just because I know that they are all pushing to try and get things up and running. But
yeah. So some of it isn't particular to the company, right? The academic centers that they're negotiating with have to go through negotiations, contract negotiations.
Oh, yeah. That's all. It's in fact all, within the hands of the centers right now. They're going through their regulatory processes,
right.
Contracting processes, things like that. But yeah, they're all diligently plowing forward.
Right, and for some sites that's easier and more quickly happening, and other sites are a little bit slower on the uptake. So, and that's probably a lot of other things we don't understand on the academic centers and what they're going through. So hopefully by fall we'll see these all up and running. So Craig, to date you've treated You have 3 patients in your study that have been actively enrolled. Can you give us a ballpark about how many others are actively enrolled in the study?
We have 8 patients scheduled for screening. I believe we have 4 randomized. We have 3 that have been dosed, and we have a large list of patients that are interested that we're diligently working through right now. At some point we will probably possibly cap out our enrollment at our site at probably around 14 patients, just because we don't want one site saturated with too many patients. We want these patients geographically dispersed. But it's been really, I think, there's a lot of interest in the Duchenne community with regard to this trial.
Right, thank you. And one last question that has come in. This is an allogenic cell product. Do you need to do immune suppression? You aren't doing it, I know. Anyway, they're immune suppressed on steroids, but are you doing further immune suppression based on the cell-based therapy?
No, there's no need for that. Right.
All right. I think that we've captured this, and we've gone about 10 minutes over. I can't thank you enough, Craig. This was terrific. Deborah, you do a great job, and Linda, thank you for explaining the basis and the rationale for using these cells. We're really delighted about this study, delighted about who you're going to enroll in the study, and the fact that this is, in terms of a trial, less burdensome trial, and hopefully more exciting in terms of, not than all the other trials, but more exciting in terms of less burden on the patient in terms of coming and receiving the dosing and so on. And certainly a shorter trial that should be facilitated. At the end of the day, we're looking for positive results. So I thank you. I'm very hopeful, and I appreciate all you're doing for us, and we all wish you Godspeed and thanks. Thank you both. Thank you all. Thanks, Linda, Deborah, and Craig, and we'll speak to you and see you soon.
Thank you, Pat.
Thanks.
This concludes the webinar. Thank you all.